Hello, I'm Dr. Joseph Shahadi and welcome to our office. Today I'd like to share some exciting news about exosomes, which is a new treatment, as well as some of the background science behind exosomes. We use exosomes along with our other stem cell treatment options here at Cedar Stem Cell Institute. You might ask yourself, what exactly is an exosome? I'd like to start off with the definition. An exosome is essentially a very, very small intracellular vesicular packet of information that contains proteins, RNA, DNA, and microRNA, which essentially gets excreted from one cell to another and helps with intracellular communication along with some other features. When I say small, I mean nanoparticle level, in the range of 30 to 100 nanometers in size. This slide is somewhat busy, but it's essentially a magnification of a single exosome over here to the right. On the left-hand side, this is a cartoon illustration of a cell that's excreting exosomes from its cell wall. That's what those small orange dots represent. And then on the right-hand side, this is a large magnified view of a single exosome, and I'd like to draw your attention to 3 or 4 aspects of the exosome. First, that it contains some DNA particles, some RNA particles, some messenger and microRNA particles, which are very helpful, which you will see later in the talk, as well as some cytoskeletal proteins such as actin and myosin that help with muscle. And lastly, the fact that there's some components that affect the immunoregulatory molecules and help with the immune system. So those are 3 or 4 points I'd like to make regarding content of exosomes. Next, I'd like to talk about the favorable characteristics of exosomes in general. First of all, exosomes last long in the blood circulation. Secondly, they're very biocompatible. Thirdly, and very importantly, exosomes can cross the blood-brain barrier, making them very useful for treating neurological diseases. And that also means they can be given intravenously instead of into the spinal fluid directly, which is less painful. In addition, exosomes have a bilipid membrane that protects the content of the exosomes, and that protects them from being degraded right away. And lastly, exosomes have the ability to be targeted specifically to organs or tissues such as the brain, the lungs, the liver, the kidneys, or the musculoskeletal system in general. Further on the functions of the exosomes, in particular with the nervous system, which is my special interest, is that, as I just mentioned, they can be given intravenously because they cross the blood-brain barrier. Secondly, they communicate or help communicate between various cells, and in particular, there have been research and evidence that suggests that exosomes are helpful in helping repair and maintain myelin sheath, which is also known as the insulation of nerves, kind of like the insulation on a wire or a cable. And that's typically the problem in multiple sclerosis. In addition, exosomes have been known to help eliminate waste, such as amyloid protein buildup found in Alzheimer's disease. And lastly, exosomes have been shown to restore the blood supply in patients who just underwent a stroke. So how do we acquire or get exosomes? Exosomes are very, very small particles, like I mentioned, in the nanoparticle range, and they're found within stem cells themselves, also known as a subcellular component, shall we say, of stem cells. And there's approximately 300 to 1,000 exosomes found in each and every stem cell. So you just have to think of it as something that's very, very small and a part of a stem cell. Exosomes are purified from mesenchymal stem cells using a very high-speed centrifuge. Exosomes are supplied by many companies, including Vidi Labs and Organocell. Here's a picture of the ultracentrifuge that spins approximately 100 times faster than a usual or average centrifuge machine. I'd like to talk a little bit about the history of the science of exosomes. This dates back almost 40 years to the early 1980s, so this is not exactly brand new. Exosomes were first described as small vesicles that were released by various cell types with a diameter between 40 and 1,000 nanometers. That's, like I mentioned, nanoparticle level. However, a little bit more recently, in 2006, it was discovered that exosomes were actually able to communicate via RNA particles from one cell to another, which was a newly discovered property of exosomes, which was very exciting. In other words, exosomes, which were formerly known as basically a garbage can or a waste paper basket for the stem cell, is now recognized to be a major player in communication. It's kind of like being thought of as a garbage man in the past, and now you're the FedEx carrier with information that's useful and targets to achieve. Here's a picture of the exosome content, also known as cargo. Exosomes contain cargo or content, and the content is complex and heterogeneous or mixed, and it includes proteins, genetic information in RNA and DNA, and this information is very important in helping one cell communicate to another cell to call it into action, if you will, to help promote healing and regeneration of damaged tissues in a complex manner. Exosomes are a very hot topic in regenerative medicine of recent. And as you can see, if one were to do a literature search online on PubMed, which is the main website that contains or holds essentially all or nearly all research articles throughout the world and in the United States. If you type in exosome as a subject matter, which I just did today, you will see over 14,000 results, which is an enormous amount of interest and research. And to the left, we can see a graph illustrating the exponential growth and interest in exosomes. So much so that these exosomes and research institutes are publishing their outcomes and their clinical research in very reputable journals such as Neuron, Lancet, the New England Journal of Medicine, Stroke, Journal of Neurosurgery, which is our main journal as a neurosurgeon, as well as journals such as Stem Cell. Thank you very much for your time and your interest in this exciting topic. For more information, please visit our website at cedarsci.com. Thank you very much.